Our final talk for this session before we head for lunch is Amro Hamdoun, who is at the Scripps Institution of Oceanography. As I said, this session is definitely heavily UCSD-centric. And Amro is also one of the co-PIs in the ADC NICE initiative, and he's always had these spectacular images of sea urchins on Twitter, so if you don't already follow him and to see these spectacular images, you should definitely go check his Twitter out. And over to you, Amro.
Thank you. Thanks for that introduction. That confirms that I have one follower. That's great. Oh, thank you.
Though
okay.
I quit Twitter, so I
okay.
maybe.
I'm on Blue Sky. Come back. I'm on Blue Sky as well.
Yeah.
Okay. So I'm Amro Hamdoun. I'm a professor at SIO. I'm co-director of this Allen Discovery Center for Neurobiology and Changing Environments, along with Martine Tresgueras, who's here, and Didi, who you heard from earlier today, and also of a related center on oceans and human health. Now, the overarching theme of our center is to understand how a changing ocean environment is going to affect marine organisms and how the nervous system may play some role in regulating that. Now, before I begin, I just want to amplify on a theme that Didi already raised, which is this idea that most of biology is studied in this small handful of animals, and they don't really represent the broad range of diversity that exists in the environment. And studying a wider range of animals, which is part of the mandate of this center, is actually not just something that you want to do for curiosity, but it actually underlies kind of many of the fundamental understandings of neuroscience that we talk about here. So Didi already alluded to the action potentials and squid and the biochemistry of memory. Sodium potassium ATPase was discovered in crabs, and we heard a beautiful talk from Eve this morning about her work on crab neural circuits. The green fluorescent protein, which all of us use, comes from jellyfish. Phagocytosis was described by Ilya Mechnikov, a Ukrainian scientist actually, in sea stars, and of course cyclins were described in sea urchins. And as you already heard, my favorite critter is the sea urchin, and many of you know this maybe from your undergraduate labs or sometime in your career. The sea urchin is a classic model in cell and developmental biology. Their superpower is that they make lots of egg and sperm, and you can fertilize them externally and nothing more than a dish of seawater. They develop perfectly synchronously, they're optically transparent, and so they've been the basis for a lot of studies of the regulatory networks that control development and for the biochemistry and cell biology of fertilization and early development, and of course the discovery of the cyclins that I already mentioned. Okay, so sea urchins are also very ecologically important, so some of you may have seen these headlines about zombie urchins. What this refers to are urchin barrens where urchins have overgrazed the kelp forest. The reason for the overgrazing of the kelp forest is that the predatory sunflower sea star actually was almost went extinct, and that was caused by, of course, climate change and disease, and these climate-induced boom and bust cycles have a major impact on the ecosystem, the kelp forest ecosystem. Now, sea urchins move slowly, so that has, incorrectly caused a lot of people to think that they're not interesting, and sometimes I get frustrated because cephalopod people brag that their organism has 8 whole limbs. 8, woo, big deal. Okay, sea urchins have a lot more than 8. And I just want to show you that they actually do some interesting things. They are not dumb rocks with spines. They're really very cool critters. So they have a writing response. So you're seeing in this movie here, they're going to help their buddy get flipped over. They're really worried about him. Okay, that's very good. They're decorated with chemosensory receptors and photoreceptors on their surface, and they have very clear food preferences. So in this movie, we give a sea urchin a little piece of dried fish, and they really like dried fish, and he's already eating a vegetable. He's eating a piece of kelp, and then he tastes that there's, or she tastes that there's a little piece of fish there, and immediately spits out the vegetable and says, here, you take the veggies, I'm gonna focus on the yummy piece of fish. And of course, sensing these different environmental cues is very important for their biology and their life history transition. Sea urchins undergo this very dramatic body plan reorganization at metamorphosis, where they go from swimming in the water column to settling down on the benthos. And in that body plan transition, they go from being bilaterally symmetric to pentaradially symmetric. And of course, that's all governed by being able to effectively sense the appropriate cues for the time and place to settle. Now, even though these animals have this complex array of photo and chemoreceptors, and they coordinate movements of dozens of independent podia, they're relatively understudied in neurobiology. They're not ideal for electrophysiological recordings like a squid or maybe a crab because they have a relatively small nerves. We know a little bit about from immunochemistry studies about the general organization of their nervous system, but we've been bottlenecked for a long time by the lack of genetics in this animal model. And most of the work that I told you about that involves early development uses transient genetic methods. Transient genetic methods have a limitation in that they only last into the early larval stages of this animal's life cycle. And what that means is that most of nervous system development, that dramatic reorganization at metamorphosis, and even some of the reproductive phases are really inaccessible by genetic tools. So we set out about 5 years ago to kind of think about, can we rethink how we work with sea urchins and build a genetically enabled urchin model? And ideally what we want is an animal that has small size, has the superpower of making lots of gametes, doesn't have some special culturing requirements, and has some good pre-existing background of knowledge. Now, there are many sea urchins used in the field, so when you talk to people about sea urchins, they often use the one that's closest to them. And the major one used was, for a long time, S. purpuratus, the purple sea urchin. And this was one of the first marine invertebrates, in fact, to have a fully sequenced genome. But many of these species have one very big problem in that they have very long egg-to-egg times or life histories. So we settled on this species. This was actually first proposed by Ralph Heingardner, an immunologist in the 1960s. And he was interested in developing isogenic lines of sea urchins for allorecognition studies. And Lidocynus pictus have a really nice adult size for culturing in the lab. They're a little bit smaller than purpuratus, yet they make a bigger and more transparent egg. They have these beautiful optically transparent larvae. And really the most important thing is that they have one of the shortest generation times of any of the widely used sea urchins. They've been used for a long time. They were actually part of the initial studies that led to the discovery of cyclins. They were the first sea urchin in which cis-regulatory reporters were described, but my favorite thing that's been done in sea urchins is that they've been to space, right? And they went up in 1996 in the Space Shuttle Endeavour mission STS-77. That was to study the effects of microgravity on cell division. And this is very important. And the reason that this is very important is, of course, this. You can be very proud if you work on a sea urchin, right? So, okay, so their larval development is very rapid. It's about 2.5 weeks. We can get them through very quickly. That turns out to be very important for making a transgenic sea urchin. The little juveniles look like this. They're very cute. We can put them out on a Petri plate and sort them. We move them around with just a little paintbrush. At 3 weeks, they transition onto what we think of like a more solid diet or macroalgal diet. So here they are kind of rushing to have their first kelp feeding. And we can grow them, just to give you a sense of scale, this is a 6-liter volume, and there's 11 animals in there. Those are sexually mature animals. So you can grow them in these kinds of rack systems that are really commonly used. So when we wanted to make a transgenic animal, we were inspired by work that was done in other systems historically. And we think about how most animals have made the transition to transgenesis. Typically, the first step is through some kind of random integration method, often a transposase, so in zebrafish, toll-2, for example. And one of the challenges in doing this in some marine animals, especially those like sea urchins that have a very dramatic metamorphosis, is that oftentimes non-stable integrations will be cleared at metamorphosis. And that had historically led to a lot of false dawns where people would put something in, and so you see in the absence of a transposase, they'd put something in, they'd get great expression off of the transgene that's non-stably integrated, but when they go through metamorphosis, those are all cleared out. Having a very fast life cycle in this animal, we were able to screen through a lot of different conditions and kind of settle on a good strategy to generate stable transgenics. We published the first F1 transgenic echinoderm last year, and we were really pleased that the transgene integration was stable, which means it persists all the way into the development of the nervous system, which becomes then the tool that we can use to label transgenes. And we've started to make different color lines of sea urchins that we can then cross back to one another to study nervous system development and function. All right, so we can do some transgenesis in this animal. That's really exciting, but how do we know what to target? So one of the things that we've been doing within ADC NICE since we started 5 months ago is really accelerating the pace at which we discover neural targets. Now, there are 2 ways that most of you probably think about doing this, and one is by in situ hybridization, and the other is maybe by some kind of in silico approach or single cell or single nucleus sequencing. Now, both of these have some limitations. Typically, in situ hybridization is this very throughput limited kind of approach, and often these sequencing approaches can identify a lot of different populations, but you can't do many different conditions. So we developed something which we're just calling high throughput HCR. So this is just standard hybridization chain reaction, but we've miniaturized the reactions. So they're about 1/10 the volume of a normal reaction. We use the sea urchins as little beads. We basically fix them, we can pour them in, we plate them out in an automated way. The entire platform, all the handling steps happen on the plate. So I know for a lot of the students that are doing this, this is nice. This is a general purpose liquid handler, so it's just an Opentrons. So you can set this up in any lab. And you can actually check it out and see. So we do 192 targets in 32 hours. And our outputs are out, and we're starting to think about how we will make this sort of accessible. But you can already go ahead and see if your favorite gene is there. We use this to set up a neural screen to find 70 or so neural or neural adjacent targets. These include things like neurogenic transcription factors. Here are 2 in particular. The imaging data that we get from this is very high quality data because it's a spinning disk confocal platform. So this is an already known forkhead transcription factor, FOXQ2, that is involved in setting up the apical organ and the early development of the serotonergic neurons, and also some unexpected candidates. So this was a gene that we found in the genome that's homologous to EMX, which is involved in vertebrate brain development, which seems to be expressed in this unexpected pattern. We've also used that now to kind of motivate discovery of things like pan-neuronal genes, which we will use to discover promoters to drive expression of things like GCAMP or other reporters, KABLAM, in these animals. And we're also starting to get a little bit of a sense of the expression of things that are unique to certain cell types that might be involved in some kind of chemosensory biology. These include things like the neuronal acetylcholine receptor that's expressed only in the tips of the 2 feet that we think might be important for some kind of chemosensory function, as well as some of the neurotransmitter transporters like SLC18A2. And we're starting to build this out into kind of a small a sort of atlas. All right, so I want to end there and just give you a sense of kind of what we've done over the last few years. Obviously, the North Star for all of this is going to be to share these resources. We don't envision that we're going to be able to do everything with the sea urchin, so we're also setting up platforms for cryopreservation of sperm so that we can send it to the sea urchin community and have other people do experiments with it, and also using some of our platforms for de-risking some of the animal species in the ADC NICE. I just want to thank, there are a lot of people that did this. As you saw, I talked about five years of work that went into those slides, and of course the Paul G. Allen's Frontiers Group and Family Foundation for supporting the new ADC NICE. Thank you.
Thanks, Amro. We can probably take more questions since we have a little bit of time. Again, I urge early career researchers to ask some questions. Okay, maybe I can start. Like so,
Yeah.
so these sea urchins, are they also like calcified in some way? They must have the spine. I've only seen like spiny
Yeah,
ones.
so they have an endoskeleton. They have a thin layer of epidermis sitting over a hard spine. Yeah.
Okay, and that spine, is it? I'm guessing it's probably going to be affected by acidification. Has anyone tested that, or?
Uh, um, yeah, people speculated about that. I'm actually a little bit skeptical that it will be affected by the range of environmental conditions that they'll encounter in nature. So one of the features of sea urchins is that they have huge population sizes and they have large numbers of offspring within each spawn. And there's some beautiful work by Melissa Pespini, who's was a postdoc at Hopkins and now is a professor at the University of Vermont. And she showed that even within a single spawn of sea urchins, there's enough polymorphism to cover a very wide range of environmental conditions. So I think things, the initial ideas about how climate might affect sea urchins might not actually hold up. Mm-hmm. Yeah.
Okay, yeah, go ahead. Sorry, introduce yourself, please.
Yes.
Yeah.
Hi, I'm Christine Merlin from Texas A&M University. I have a technical question. I was wondering whether you know why Minos transposon are actually working and not TOL2 or piggyback. And I'm asking because it seems to be very similar to what we see in butterflies.
Yeah, thanks for the question. It's funny, when we started, and in fact, this is one of those examples where for the PIs in the room, you should always listen to the people in your lab, because I said TOL2 is going to work. And because everybody, I have friends that do zebrafish, and they said, TOL2 is great, it works too well. How could TOL2 not work? I've been talking to people that do this in other systems, other marine systems, and they found a similar pattern where they're having trouble with TOL2 and piggyback. It could simply be that we did something wrong. There's some condition we can optimize to make TOL2 work, but in our hands, we didn't get any integrations. And in fact, some of the earlier groups that had much more difficult pathways towards screening out for stable integrations started with TOL2 and piggyback, and when they didn't see anything, just stopped. So I think it's really, the lesson there, if you're going to try to de-risk an animal, make sure you can screen a lot of different transgenesis methods, because it's hard to know a priori which one's going to work. But yeah, and I don't know why. So the short answer to your question is I don't know why.
I kind of want to supplement that answer a bit. I think maybe some of it depends on whether it's vertebrate versus invertebrate systems. And besides just testing a whole bunch of stuff, I also think that there are ways you could probably experimentally evolve versions of these tools that work well in your system. I'm happy to chat about that offline as well. But yeah, just taking something that works in zebrafish or the standard model systems and putting them into non-model organisms is not easy or straightforward.
Yeah, I didn't show the data, but one other thing to consider is that there's some threshold of efficiency that you need. So, and that depends on the fecundity of your animal and how many you can move through. Once you hit that threshold of fecundity, you might be done optimizing, and it's time to move on and just start making what you need to make.
I'm Noah from the MVL. I was wondering if you've thought about CRISPR much. Is there a technical barrier there? Those large transparent embryos look.
Oh yeah, okay. Can I show a slide?
Yeah.
Oh, okay. I might have a slide on that. Okay. Let me see if I might have a slide here that, okay. Yeah, we've thought about CRISPR a lot. So let me see if I can just put this all up. Oh, what happened? Oh, see, this is good. This lets me go over on time. Okay, so there are a lot of ideas about how to make CRISPR work in unconventional models, and we've tried a lot of them. One of the things that you've probably heard about is biotinylating arms, single-stranded arms, long homology arms, short homology arms. And no matter what we tried, we could not get efficiency to an acceptable level. And our efficiencies hovered around 2%. And we just took that personally for a long time and felt bad about ourselves. And then luckily, one of my colleagues published a paper in sea urchins showing that they were getting very similar low efficiency with a wide range of methods. And so we decided to go all the way back to the drawing board and think about, well, what governs efficiency in something like a CRISPR method? And one of the top factors that governs efficiency, especially in a rapidly dividing cell like a sea urchin embryo, is the size of the cargo that you're trying to integrate. So we were inspired by some work in zebrafish in which what you can do is you can use your transposase to integrate most of the fluorescent protein under control of a ubiquitous promoter. In this case, we use the polyubiquitin promoter to drive most of the fluorescent protein in all cells. And then what you do is there's a small complementing piece of the split fluorescent protein that you integrate by CRISPR, and you integrate only that small piece in your cell type of interest. And so the strategy we're using now is basically to make a transgenic line in which most of the fluorescent protein is already integrated, and the cargo that we have to deliver by CRISPR is much, much smaller. And what you can see is that that gives us a very dramatic increase in the efficiency of integration. And that gets us up to the 25–30% level, which is enough to effectively then raise animals up and get germline integrations and make your line. Yeah.
Yeah, Wolfgang. Yeah. So in the meantime, I also wanted to supplement that and say that I think there's another idea that you can use, because if you need to make knock-ins, the thing is you need to facilitate HDR in some way. So, but there's also a lot of competing NHEJ. So, I think it's also useful to try and suppress that NHEJ transiently so that you can maybe increase the efficiency of an HDR just by chance. And there are some drugs that do that. It's worth testing.
We've tried those. In our hands, we haven't had much luck with that. But again, that may be a feature of the fact that we're working with a very rapidly dividing cell that has a very short interface.
Mm-hmm.
Yeah.
Yeah, go ahead.
Yeah. Wolfgang Stein from Illinois State. I'm wondering about the writing response that you showed at the beginning. Do they have a staticist or something? What's the sensory organ that allows them to do this? Do you have any idea?
I don't know. Yeah. It's been used for a long time in the field. I don't think, to my knowledge, it's not known, but it's been used for a long time as like the standard behavioral assay. There's one other that's used, which is if you take sea urchins and you immerse them. So you put them, imagine you put a beaker on top of a small volume of water and the beaker's out of water. They'll immediately figure out the water's below and go down. If you repeat the assay over and over again, over time, they'll go down much faster. So once they learn which direction to go. Maybe learn's a strong word, but once they're conditioned. Yeah.
Cool. If there are no further questions, let's thank all the speakers from this session.